So hello and good morning and good evening, everyone. My name is Junken Aoki from the University of Tokyo in Japan. So first of all, it's my great honor to give you my lecture on lipids. And I would like to thank organizers for giving me this nice opportunity. I would like to start with my lecture on the structure and functions of glycerophospholipids. Today, I would like to talk the following topics as in the slide. First, I'm going to talk about the structure of glycerophospholipids. Second, I will explain the function of glycerophospholipids, especially focused on bioactive lipids derived from these glycerophospholipids. In particular, I'm in the field of lysophospholipid mediator. I will explain detail about the lysophospholipid mediator. I will also mention special types of glycerophospholipids, such as inositide and oxidized glycerophospholipids. And in the second half of my lecture, I will explain the biosynthetic pathways of glycerophospholipids. This includes the de novo pathway and the remodeling pathway. I will also discuss about the enzyme involved in the remodeling pathway. And finally, I'd like to introduce our recent study on the remodeling of SN1 fatty acid in glycerophospholipids. So there are many types of lipids in the cells, and these include triacylglycerol, glycerophospholipids, cholesterol, and glycolipid as shown here. And today in my talk, I'd like to focus on the structure and function of glycerophospholipid, such as PE and PS. This slide shows a schematic drawing of glycerophospholipid molecule. Roughly speaking, phospholipids are divided into two parts, polar head and hydrophobic tail. If we look more closely at the structure of glycerophospholipids, we can see that the majority of glycerophospholipids in the cells are composed of a polar head with an phosphate group attached to the alcoholic substances as shown X and two fatty acids. I have to mention here that there are some glycophospholipids that cannot be described by this formula. These are, for example, bis monoacyl glycerophosphate, BMP, or lysophosphatidic acid, and cardiolipin. Both BMP and cardiolipin have a glycerol in their structure and grouped into glycerophospholipids. Although today I don't explain them deeply, BMP and cardiolipin are rich in lysosome, late endosome, and mitochondria, and have a specific function in these organelles, respectively. Thus, there are some exceptions to this formula, but the major glycerophospholipids detected in the cells are composed of the three parts, polar head group, and glycerol backbone, and two fatty acid moieties. At the polar head, we can see many substances with a hydroxy group. Those are attached to the phosphate group by a phosphatidic ester bond. These include choline, ethanolamine, serine, inositol, various phosphorylated form of inositol, glycerol, hydrogen, glucose, threonine, cytidine, phosphate, etc. Thus, glycerophospholipid molecule with more than 20 polar head group exists in the cell. I think the development of lipidomics technology may lead to the discovery of novel phospholipid with previously unknown polar head group in the near future. Second portion, fatty acids. The major phospholipid in the cells have two fatty acids. This fatty acid binding sites is called SN1 position, and this fatty acid binding site is called SN2 position. As shown in this slide, the fatty acid found in phospholipid in the cell differ between the SN1 and SN2 position. The SN1 position is occupied mainly by saturated fatty acid, such as palmitic acid C16O, stearic acid C18O, and monounsaturated fatty acid oleic acid C18-1. On the other hand, the SN2 position is mainly occupied by unsaturated fatty acids that have multiple double bond of the cis type, as shown here. So it is important to note here that we can detect glycerophospholipid containing only saturated fatty acid or containing only unsaturated fatty acid in both positions, as shown here. So about at least 10 fatty acid species are linked either at the SN1 and SN2 position in the glycerophospholipid in the cell. So glycerophospholipids also show the diversity in the binding of fatty acid to the glycerol backbone. As shown here, there are at least 3 patterns in the binding mode of fatty acid to the glycerol backbone. This creates diacyl glycerophospholipid, alkyl glycerophospholipids, and alkenyl glycerophospholipids. Thus, phospholipids are composed of about 20 different head groups and at least 3 different head backbone, and at least 10 different fatty acids, both at the SN1 and SN2 position. Therefore, in theory, glycophospholipid species may consist of more than 1,000 species, as shown in this slide. And I believe the elucidating the biological significance of this diversity of glycerophospholipid is one of the challenges in our current lipid biology. Here, I would like to show you a new technique to visualize the distribution of phospholipid species on this section. This new technique is called MS imaging. This allows us to visualize each of phospholipid molecule species on the tissue sections. Detail of this technique will be explained in more detail on my lecture tomorrow. So, for example, this slide shows the expression of specific glycerophospholipid in mouse tissues. These analysis, from these analysis, we knew the molecular species glycerophospholipids are expressed very differently in the mouse embryo. For example, as you can see here, 3 phosphogylyl ethanolamine PE species, 34 to 46 and 38 to 4 PE species showed very different cellular distribution pattern in the mouse embryo. The same is true for the 3PS hospitalizing species shown on the right. So these analyses strongly indicate that the glycerophospholipid species are expressed in cell-specific manner. So it is also likely that lipid molecules with the same structure have tendency to aggregate by themselves. Thus, each glycerophospholipid species may form a lipid domain on the cell membrane rather than existing in a random manner, as in the mixed model shown in this slide, as shown here. The molecular species of glycerophospholipids may define the membrane protein that accumulate in each domain. Thus, understanding the diversity of glycerophospholipids is considered to be the major theme in lipid biology. The diversity of glycerophospholipid species is more complex when we consider the fatty acid oxidized. In general, polyunsaturated fatty acid in phospholipid molecules are known to undergo per-oxidation reaction. This results in the production of a variety of oxidized phospholipids. Let us consider the per-oxidation reaction of linoleic acid C18-2, which is the most simplest polyunsaturated fatty acid. As shown in this slide, the two double bonds of linoleic acid can be attacked by reactive oxygen species loss at the site indicated by the arrow, which results in the production of 31 different peroxides, including the four structures shown in the diagram. Thus, the oxidation of fatty acid in glycerophospholipids leads to an exponential increase in the number of phospholipid molecular species. Recent improvement in MS mass spectrometry techniques have revealed that there are many oxidized phospholipids in our body. So elucidating the function of oxidized phospholipid is one of the current challenges in lipid biology. So we would like now to turn to the functional aspect of glycerophospholipids. As with other lipid molecules, glycerophospholipids are thought to have four basic functions as component of cell membrane, as protein modifier, as in the case of glycosyl phosphatidylinositol-anchored protein, as nutritional molecule. Example is choline supply. And glycerophospholipids can also function as cell signaling molecule and their precursors. First, the glycerophospholipids are a major component of the biological membrane, and they account for about half the content in the cell membrane in erythrocytes. In some membranes, such as the myelin membrane, lipid molecules make up more than half of the membrane. Thus, the primary function of glycerophospholipids appear to be the component of cell membrane. In addition, glycerophospholipids also have important role in cellular signaling. Phosphoinositides are phosphorylated phospholipids in which the 3, 4, 5 position of the phosphatidylinositol hydroxy group are phosphorylated. They are deeply involved in cell signaling. For example, PIP2, which is a phosphoinositide phosphorylated at the 4 and 5 positions, is degraded by phospholipase C beta to form diacylglycerol and IP3, which then contribute to the activation of PKC and calcium signaling respectively. Phosphorinositide PIP3 phosphorylated at position 3, 4, 5 of the inositol ring act as a docking site for intracellular signaling protein with pH domain, such as AKT, and are important in intracellular signaling. Glycerophospholipids are also important as precursors of lipid mediators. To produce lipid mediators, glycerophospholipid precursors are subject to various phospholipases and oxidative enzymes. These lipid mediators are mainly recognized by G-protein coupled receptor, which in turn trigger a variety of cellular responses. Such lipid mediators, including prostaglandin, leukotriene, 2-arachidonoglycerol, anandamide, and lysophospholipids. Prostaglandin and leukotriene were nicely explained by Dr. Ennis in his talk last night. I would like to talk mainly about lysophospholipids. As shown in this slide, a variety of lysophospholipids can be found in biological fluid, such as plasma, serum, saliva, urine, and cerebrospinal fluid. These lysophospholipids include lysophosphatidyl LPC, lysophosphatidyl LPE, lysophosphatidyl LPI, lysophosphatidyl LPG, lysophosphatidyl LPS, lysophosphatidic acid, LPA, and sphingosine-1 phosphate, S1P. Of these, LPA and S1P is the first to be analyzed as lysophospholipid mediators. Of course, S1P is single lysophospholipid and is not a glycerophospholipid. So I will focus on glycerolysophospholipid. In the past 2 decades, several GPCRs have been reported to respond specifically to LPA, S1P, lysoPS, lysoPI, lysoPG, and lysophosphatidyl glucose. I have been working on LPA signaling. So first, I would like to talk about LPA first. So 6 GPCR that specifically respond to LPA have been identified, and they are known, now known as LPA1 to LPA6 in the order of discovery. LPA is the simplest glycerolysophospholipid, consisting of 1 glycerol backbone, 1 fatty acid, and 1 phosphate group. In general, LPA with an unsaturated fatty acid attached to the SN2 position of the glycerol backbone is more biologically active, suggesting that some kind of phospholipase A, PLA1 is involved in the pathway of LPA production. So far, at least 2 pathways of LPA productions are known. In one pathway, lysophospholipid, such as LPC, is converted to LPA by secreted enzyme named autotaxin with its lyso-PLD activity. In another pathway, phosphatidic acid PA is converted to LPA by an extracellular PLA1 specific to PA. This enzyme is known as phosphatidic acid selective PLA1, PA-PLA1 alpha. PA-PLA1 alpha is also known as lipase H, and is a member of pancreatic lipase family. Very interestingly, unlike other members of pancreatic lipase family, this PLA1 alpha does not show lipase activity to hydrolyze triacylglycerol, but shows PA-specific PLA1 activity producing SN2 type LPA. Analysis of knockout animal as well as human genetic disease of these 6 LPA receptors and 2 producing enzyme uncover the role of LPA signaling as physiology. For example, LPA produced by autotaxin is involved in embryonic blood formation via LPA4 and LPA6 receptor, since ATX knockout mice and LPA4 and 6 double knockout mice are embryonic lethal and show similar angiogenic abnormality. LPA produced by autotaxin is also involved in implantation of fertilized egg in the uterus via LPA3 receptor, and also engaged in chondrogenesis via LPA1 receptor. LPA produced by PLA1 alpha is also involved in the formation of hair follicle by LPA6 receptor, as shown here. So in the past decades, novel GPCRs that specifically react with glycerol lysophosphoripid had been identified. These include receptors for sugar containing lysophosphoripid and serine containing lysophospholipids. For example, GPR55 is activated specifically by sugar containing lysophospholipids, such as phosphatidyl inositol, LPI, phosphatidyl glycerol, and glucose containing lysophosphoripid, LPGLC. Although their detailed functions are not yet known, recently, several GPCRs that respond specifically to oxidized phospholipid have been also identified. These are members of mass-related GPCR expressed specifically in DRG neuron. So through the functional analysis of these GPCRs, the function of lysophosphatidyl lysophosphatidyl inositol, and oxidized phospholipids may soon be elucidated. Now, I'd like to turn to the biosynthetic pathway of glycerophospholipids. In order to understand the significance of the diversity of glycerophospholipids, as I mentioned, it is necessary to understand the molecular mechanism by which the diversity is formed. In this section, we will describe the biosynthetic pathway of glycerophospholipids, especially the Kennedy pathway and the Lange pathway, which was proposed in 1950s. The enzyme involved in this pathway will be also discussed. And finally, as a research topic, I will talk about the enzyme involved in the remodeling of fatty acid SN position, fatty acid at the SN1 position of phospholipids. First, I would like to look at the outline of the biosynthetic pathway of glycerophospholipids. As shown here, glycerophospholipids are produced mainly 2 pathways, the Kennedy pathway and the Lange pathway. The Kennedy pathway is also known as a de novo pathway, in which glycerophospholipid molecules are newly synthesized. In this pathway, glycerol 3-phosphate G3P, derived from sugar metabolism, undergoes a 2-step acyl transfer reaction to convert it to phosphatidic acid, PA. Then PA is further converted to diacylglycerol, DAG. PA is also the PA and DAG in turn is converted to phospholipids such as PC, PE, and PS and PI by head exchange reaction. Let's take a closer look at the enzyme involved in this Kennedy pathway. This reaction involves the enzyme introducing fatty acids and enzyme exchanging the polar head. GPAT, glycerol-3 phosphate acyl transferases, derived acyl transferase, which catalyzes the reaction of G3P to LPA and LPA-AT, lysophosphatidic acid acyl transferases, which catalyze the reaction of LPA to PA as an enzyme introducing fatty acid. Although I don't explain the head exchange enzyme in detail, several enzymes have been identified and characterized. Through these pathways, various glycerophospholipids are de novo synthesized. This slide shows a phylogenetic tree of acyl transferases. Acyl transferases are the enzyme that transfer fatty acids, and there are about 20 different acyl transferases belonging to either the M-BOAT or AG-FAT families. 4 GPAT molecules and 4 LPA-AT molecules have been identified. And as you can see here, both AG-PATs and LPA-ATs belong to the AG-PAT family and form a cluster in this tree. Next, I will talk about the enzyme involved in the remodeling pathway. First are the enzymes involved in the remodeling of fatty acid at the SN2 position of phospholipid. This reaction proceeds in 2 steps. The first step is catalyzed by phospholipase A2, which removes the fatty acid at the SN2 position. And the second step is catalyzed by SN2 type lysophospholipid acyl transferases, SN2 LPLATs, which introduce the fatty acid at the SN2 position of SN1 LPL. More than a dozen of isozymes have been reported as candidates of PLA2 molecule. And as an SN2 LPLAT, 2 LPLAT molecules that specifically introduce arachidonic acid were identified. In this phylogenetic tree, LPC83 introduce arachidonic acid into the SN2 position of phosphacylcholine, and other enzyme MBOAT7 introduce arachidonic acid into the SN2 position of phosphatidylinositol. Including these LPA83 and MBOAT7, more than 10 kinds of lysophospholipid acyl transferase molecules have been reported, belonging to either MBOAT family and AG-PAT family. Next, I will talk about the enzyme involved in the remodeling pathway. First, I'd like to talk about the enzyme involved in the remodeling of fatty acid at SN2 position. Sorry. So finally, I'd like to talk about a recent topic, the molecular mechanism of fatty acid remodeling at the SN1 position of phospholipids. In the 1950s, Rands et al. Reported that not only SN2, but also the SN1 fatty acids are remodeled. Similar to the remodeling of SN2 fatty acid in phospholipids, the remodeling of SN1 fatty acid proceeds in 2 steps. The first step is removal of SN1 fatty acid by PLA1. And in the next step, fatty acids are introduced into the SN1 position of the resulting SN1 type lysophospholipids. However, the nature of the enzyme involved in the remodeling with SN1 position has remained to be solved. In particular, molecular detail of both PLA1 and the SN1 LPL80s have been not well understood. In this slide, a commonly used lysophospholipid acid transferases as a method is shown. This reaction requires 3 components: acyl-CoA as an acyl donor, lysophospholipid as an acyl acceptor, and a microsomal fraction as an enzyme source. In this reaction, these 3 components are mixed and reacted for a certain time, and after which the phospholipids produced are quantitated by LCMS. So in the SN1 LPL80 assay, there have been a problem with the lysophospholipid used as an acceptor. Namely, preparation of SN2 lysophospholipid required for SN1 LPL assays have been difficult. In general, SN2 lysophospholipid, which contain a fatty acid at the SN2 position, are known to be very, very unstable. As shown in this slide, this instability was discovered by Dr. Ed Dennis nearly 40 years ago, and it was known that the fatty acid of SN2 lysophospholipid undergo intramolecular acid transfer reaction and are easily transferred to the SN1 position, especially under neutral and alkaline conditions. As a result of this acid migration reaction, most of the SN2 LPL preparation are the mixture of almost 90% of SN1 lysophospholipid and 10% of SN2 lysophospholipid. So it has been difficult to prepare stable SN2 lysophospholipids required for SN1 LPL80 reaction. To solve this problem, we first searched a condition in which the acid migration reaction did not proceed. The answer is very simple. By just bringing the pH of the solvent to lower pH, the acid migration reaction was completely blocked. In this experiment, PS was first digested by PLA1, kept in various pH, and resulting SN2 lyso-PS was monitored. As you can see here, in low pH, lower than 4.0, the acid migration reaction was completely blocked. So using this condition, we extracted phospholipid from mouse liver and analyzed by LC-MS. As shown in the right panel, when we use the previous method using pH 7 and room temperature condition, most of the lyso-PS with unsaturated fatty acids were detected as SN1 phospholipids, as shown in the right panel. In contrast, when we use a new method, most of the lyso-PS species with unsaturated fatty acids are detected as SN2 lyso-PS, as shown in the blue bar. So this is a protocol for preparation of stable SN2 lysophospholipids. First, we digest phospholipids with using fungal PLA1, and after 30 minutes, the reaction was stopped by adding methanol and hydrogen chloride to bring the lower pH. After the fatty acid and the remaining phospholipids are removed by C18-based cartridge column, the flow fraction was used as SN2 phospholipids. In our condition, the purity of the SN2 lysophospholipids obtained are about 95%, with contaminating only 5% of SN1 lysophospholipids. So using the SN2 lysophospholipids as an acceptor for LPL80 assay, several SN1 LPL80s have been identified. These are LPC1881, LPCat1 here, LPCat4 here, and LPC81 here, and LPG81. All are members of the GPAT family members, as shown here. Enzymatic character of the identified SN1 LPL80s were summarized in this table. LPCat1 incorporate mainly palmitic acid, both at the SN1 and SN2 position. LCL80 incorporates stearic acid and contribute to the formation of specific PI species, in addition to cardiolipin molecule. LPGAT1 incorporates stearic acid at the SN1 position and contribute stearic acid containing glycerophospholipid. Finally, LCAT4 introduced oleic acid specifically at the SN1 position. So after the identification of these SN1 LPL80s, much will be known about the biological significance of the SN1 fatty acid in near future. That's all my talk, and thank you for listening. And I'm happy to take questions. Thank you.